Hello, and welcome everyone to the COVID-19 panel webinar hosted by Bio-Rad. My name is Carolyn Reifsnyder. I'm the Global Director of Product Marketing in Bio-Rad's Digital Biology Group and your host for today's webinar. Joining me is a panel of experts I'll introduce in just a moment. First, a short introduction and a few housekeeping notes on webinar logistics. The webinar will last approximately 1 hour. Following 3 short presentations by our speakers, I'll reintroduce our panel, and we'll have 20 minutes of live questions and answers. The webinar will be available on demand after our live presentation today, and you'll be able to download the slides from today's talk, along with additional materials and web links from the resource list on your screen. Throughout the webinar, feel free to use the question and answer box on the left-hand side of your screen to submit your questions directly to us. We'll get to as many of your questions as we have time for, and if we can't get to your question today, we'll make sure to follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. An outbreak of pneumonia caused by novel coronavirus in Wuhan City, Hubei Province, China, was identified and reported to the World Health Organization on December 31st, 2019. The rapid spread of the virus to numerous areas throughout the world necessitates preparedness and response in healthcare and lab facilities. The availability of specific and sensitive assays for the detection of the virus is essential for accurate diagnosis of cases, assessment of the extent of the outbreak, monitoring of intervention strategies, and surveillance studies. In today's webinar, you'll hear about Bio-Rad's approach in partnering with CLIA labs like Biodesics, as well as information on our tests using Droplet Digital PCR and antibodies, both pending emergency use authorization. Bio-Rad has additional webinars available on demand and upcoming, outlining more offerings for qPCR, standards, and tools for research and vaccine development. Now I'd like to introduce our panel. First, Dr. Monica Herrera. Monica is the R&D program lead for prenatal testing and infectious diseases at Bio-Rad's Digital Biology Group. Next, Dr. Hestia Mellert. Hestia is the Senior Director of Development at Biodesics. Thirdly, Dr. Jean-Francois Muscadet, who is our Associate Vice President of R&D in Bio-Rad's Clinical Diagnostic Division. Also on our panel for the live Q&A session are Dr. Diana Marr. Diana leads the Applications Development Team at Bio-Rad's Digital Biology Center. Jerry Bunyaratana-Kornkit. Jerry is a Principal Scientist at Exact Diagnostics, now a part of Bio-Rad. And Stephen Blakely. Stephen is the Global Director of Marketing for Gene Expression and Cloud Software at Bio-Rad. So with that, I'm going to hand it over to you, Monica.
Wonderful. Thank you, Carolyn. Good morning, good afternoon, everyone from wherever you may be joining. In the next several minutes, I will go over the workflow and some performance characteristics for the Bio-Rad SARS-CoV-2 ddPCR test. The Bio-Rad SARS-CoV-2 ddPCR test is a reverse transcriptase digital droplet PCR test intended for the qualitative detection of nucleic acid from the SARS-CoV-2 virus present in respiratory specimens. We have validated the test on Bio-Rad's QX200 and QXDX digital droplet systems. And as you can see in this diagram here, once the RNA sample is extracted, the workflow consists of 4 steps. We begin with the preparation of the RT-ddPCR reaction. The test comprises of the triplex probe assay, which includes the primer and probe sets for the N1 and N2 regions to detect the SARS-CoV-2 virus, as well as a target against the human RNase P gene used as an internal control. We also have the reagents from the one-step RT-ddPCR advanced kit for probes, which are the reverse transcriptase, the supermix, and the DTT. And finally, we also include Exact Diagnostics standard and negative reagents, which serve as the positive and extraction controls respectively. We start by combining the assay with the supermix, the reverse transcriptase, and the DTT into each of the 96 well PCR plate. We then add the extracted RNA from the samples, and we use the controls identically as a patient specimen. We must run one positive, one extraction, and one no template control for each batch of samples run. The next step is the droplet generation. At this point, we have 22 microliters of each ddPCR ready sample in the PCR plate that has been heat sealed with foil. We load the plate onto the automated droplet generator. The instrument then uses microfluidics to combine the oil and the aqueous sample to generate up to 20,000 nanoliter size droplets that are required for ddPCR analysis. Each droplet at this point has the sample and the master mix required for ddPCR, effectively reproducing the reaction many thousands of times, which is the key characteristic that enables the high sensitivity for our test. Once the sample has been dropletized, the plate is transferred to a thermocycler to perform reverse transcription to generate cDNA, followed by PCR amplification. And finally, droplet reading and the analysis step. Once the PCR step is complete, the plate is loaded onto the droplet reader. Here, the droplets in each well are singulated and flow past a two-color fluorescence detector. The fluorescence intensity of each droplet is measured in two channels, FAM and hex, and the droplets are determined to be either positive or negative for each target within the test, N1, N2, and RP. This fluorescence data is then analyzed by Quantisoft and Quantisoft Analysis Pro softwares to determine the presence of SARS-CoV-2 in the specimen. One of the biggest benefits of this ddPCR workflow is the ability to scale up to meet high volume demands. A setup with one automated droplet generator, 3 thermocyclers, and up to 3 droplet readers can process about 700 samples per day in a single 12-hour shift. Now I'd like to go a little bit on the test configuration. Our test includes the primers and probes that for the detection of SARS-CoV-2 that are the same as those reported by CDC, and were selected from regions of the nucleocapsid gene. These are specific for the detection of the 2019 novel coronavirus. As I mentioned earlier, an additional primer and probe set is included for the human RNase P gene as an internal control. These 3 sets of primers and probes are combined into a single assay multiplex that are run in a single well per test. In the image we have here, this is a 2D plot showing the 8 distinct possible cluster combinations that can result from this multiplexing. The x-axis shows the fluorescence amplitude in the hex channel, and the y-axis shows the fluorescence amplitude in the Y channel. Clustering occurs using the Quantisoft Analysis Pro software. And we start with the triple negative cluster that we see here in the bottom left side of the plot in gray. Immediately above that cluster is a single positive cluster for N1 in the FAM channel, which includes positive droplets only for the N1 target. At a 45-degree angle from the triple negative cluster, we see a cluster in yellow, which is positive for N2 and has positive droplets in FAM and in hex, since N2 includes a combination probe of FAM and hex. To the right of the triple negative cluster, we have our RP positive cluster in the hex channel, seen here in purple. Above the N1 and N2 positive clusters, here in tan color, we have our first double positive cluster for N1 and N2, which includes droplets that are positive for both N1 and N2 amplicons. Here in light blue, to the right of the N1 positive cluster and above the RP positive cluster, we have our second double positive cluster for N1 and RP. to the right of the N2 positive cluster and at a 45-degree angle of the RP positive cluster, we have our third double positive cluster for N2 and RP, which includes positive droplets for both targets. And finally, here at the bottom right corner, we have our triple positive cluster, which has droplets that are positive for all three targets, N1, N2, and RP. So as you can see, those are all of the possible combinations of clustering that can occur. And I want to end this section by showing some of our preliminary data from our limit of detection studies. We created positive contrived samples by spiking synthetic SARS-CoV-2 virus into pooled negative patient specimens, and then we performed serial dilutions. We confirmed our limit of detection by testing 3 two-fold serial dilutions, each with 20 replicates. We determined the limit of detection to be where at least greater than or equal to 95% of the replicates were positive, meaning 19 out of 20. As you can see in the table here, for our N1 target, the limit of detection is 0.26 copies per microliter. And the N2 target, the limit of detection is 0.291 copies per microliter, which is equivalent to about 5 viral copies detected per ddPCR reaction. So I'm going to end with that and now hand it over to Dr. Mellert for her to share the Biodesics experience. Thank you.
Well, I wanted to start by saying thank you to Carolyn and the Bio-Rad team for the invitation to represent Biodesics. As well, thank you, Monica, for describing the workflow so well. It really sets us up nicely for the next portion of the discussion, which involves how this has been implemented at Biodesics in our CLIA laboratory. So Biodesics' mission really is focused on lung nodule and lung cancer diagnostics. So obviously that doesn't have to do with infectious disease necessarily, but as the pandemic has arisen, we appreciated the fact that our laboratory was in a really good place to support testing. So we have 2 locations for testing at Biodesics. The first one in DeSoto, Kansas, has specialties in high throughput ELISA as well as LC-MS, where we run a nodule risk assessment test out of. And then additionally, in our corporate office, we have additional CLIA laboratory in Boulder, Colorado. And there we focus on both Droplet Digital PCR as well as MALDI-TOF mass spec and provide testing called the lung reflex test to patients that have been diagnosed with NSCLC. So we already were processing samples for liquid biopsy. It's called the GeneStrat test, and it looks for cell-free nucleic acid for driver mutations in lung cancer using Bio-Rad's technology, and it made a lot of sense in this time to transition then to also test for SARS-CoV-2. So we initiated the efforts early March to start processing specimens in our laboratory. And so it really started out with feasibility, working very closely with Bio-Rad. We have a long relationship with Bio-Rad over the past 5 years because of our GeneStrat test, as I mentioned. And so we worked with them to onboard the process, to optimize things like extractions, and to understand our thresholds and cutoffs for the assay. And then following that, we brought the assay through the validation as is recommended from the FDA template for CLIA high complexity laboratories. We're also BSL2. So we were able to follow that pathway. And I've described here in this slide the experiments that were performed to support the EUA submission. Unfortunately, since we are in review with FDA, I won't describe the data specifically at this time, but do look hopefully in the future for that information to become available. So the process was pretty straightforward. First, we started with a limit of detection study. It was done very similarly to what Monica just described was done at Bio-Rad. So 3 different concentrations again, and looking for 95%. So that was 60 samples, 20 at each concentration. Those specimens were contrived, and the material that we spiked into negative NP swabs was sourced from BEI, which is an FDA recommended or approved source of material for these studies. Additionally, we did what they're calling a clinical evaluation study, which was also done with contrived specimens here. There were 30 that were spiked to be positive, as well as 30 negative, blinded to the laboratory, and we tested those specimens to determine the clinical sensitivity and specificity of the assay. At that time, once the validation is completed, we could also lean on the inclusivity and cross-reactivity data that's provided by the CDC, as these assays are very similar to those from the CDC. We started testing, so that happened earlier this month. And then that was upon letting the FDA know. And then within a couple of days after that, we had submitted our actual EUA information to them. So following our first clinical specimen coming into the lab, we did an external accuracy study. This was done with Dr. John Longshore at Atrium Health. And he has a EUA approved test, so we were able to do concordance of the first 5 positives and 5 negatives as well. So this is really early data. We, again, just started testing earlier this month, but I wanted to give you a taste of what it looks like in the CLIA laboratory and what we're doing to maintain control of the assay over time. As Monica mentioned, there are 3 controls that are required with the assay. So there is an extraction control, where we're using a human cell line. And there's also a positive and a negative PCR control. So this is the Exact material that we can discuss later. I know someone on the panel from Exact is with us, as well as a no template control. And so what we've set up here are control limits, and we're continuing to monitor these controls over time. So that's what you can see in the chart on the right-hand side for the first week of testing for our extraction control. So this is a really important part of on-market monitoring of the assay performance in the CLIA laboratory. And finally, I wanted to provide you with some metrics related to what we're experiencing. So we started out really focused on our community. And so we have been working directly with hospital systems in the Colorado area and are now starting to expand outside as well. So those hospitals are batch shipping us specimens, and the average turnaround time from those specimens arriving at our doorstep to being able to result back to the physicians is 34 hours, which is really two-phased. Most of the specimens go in the next day, essentially, in that 24 to 48 hour bucket. As you can see there, that's about 70% of the specimens. In some cases, when there aren't enough copies of the RP gene that Monica was referring to earlier, that's the internal control gene, we will attempt a second run. So there will be one internal rerun that occurs to try to rescue that specimen and give the patient and the physician a more actionable result. Rather than an invalid result. So there is, in small numbers, it's less than 10% at this point, we're seeing that we can't get enough material, essentially, to be able to result. Currently, our cumulative positive rate is about 17% in our community setting and with the hospitals that we're working with. And our throughput, we can get through 1,000 tests per day. We're currently running 7 days a week and on extended and staggered shifts to meet this. So we can really make sure that those patients in the community are getting their results in the fastest possible turnaround time. So with that, I thought for just a little bit of fun here, I provided some pictures of the people that are the true heroes in the laboratory processing these samples at Biodesics. So I have here some of our technical supervisors and general supervisors. You can see the box there is a shipment of batch specimens coming in at 4 degrees that will start through the processing steps. And here you can see one of our individuals in the lab working with our laboratory information management system, where she's accessioning the specimens. So that is generally the experience that we're having. Things, obviously have really picked up quickly in the laboratory, but we're really happy to be able to have an impact and support the community during the pandemic. And we're also looking forward to understanding how things are evolving. Obviously, the molecular test that looks for the virus itself is very impactful and useful in the field. In addition, we're looking forward to understanding more about what Bio-Rad's doing to test serology samples and looking at the antibody side of things. So with that, I will transition to Jean-Francois, and looking forward to hearing from him about that topic.
Thank you, Estia. Good morning, everyone, or good afternoon. So today I would like to spend a few minutes introducing you to our new Platelia SARS-CoV-2 total antibody kit, its principles. So this kit is an ELISA plate assay that detects all antibodies that are produced by the immune system upon infection in a body. As you can see on the picture on the left bottom, the immune system becomes detectable after 4 to 7 days post-infection, after the onset of symptoms, and it builds up upon time. So detecting the antibodies is a very sensitive way to detect the infection at distance away from the infection time. And so at some points, the viral load is going to decrease and may become not detectable anymore. And at that time, the total antibody, the antibody built by the immune system, is a very good way to detect the infection. Also in that test, we are detecting all the antibodies, which means the IgG, the IgM, and the IgA as well. And that's a way to increase the sensitivity of the assay, because those antibodies rises at different time, and by combining all of them, we can widen the window of sensitivity of the assay, which means that we can detect as early as 5-6 days post-infection, and that continues over time, over the whole course of infection and beyond, when the immune response has built. So on the right side of the slide, you'll see the principle of the test. So the way we detect the antibodies, we use a plate on which one protein of the virus is bound. So that's the nucleocapsid protein of the virus, which is an extremely well-conserved protein. So that ensures that even if there are some mutations, we will still detect the antibodies against that protein. So when a patient sample is get in contact with this protein, the antibodies, if there are, will bind to the protein. And then we will use another copy of that protein to detect this complex. And this second protein is bound to a reporting group. Here, this is peroxidase. So when those two proteins are linked by an antibody, we get a signal. So using twice the two copies of the protein makes the test very specific, because the antibody needs to bind to 2 copies of the same protein. So it increases the specificity a lot, as you will see in the performances. So we've been developing this test quite fast, and actually we were able to do that because we already had a test in a drawer that was a test that was developed for the SARS 10 years ago. So at that time, we were able to work out this very efficient format. And it turns out that the CoV-2 is extremely close to the SARS. It's really a close cousin with a very high homology. Proteins are almost the same. So we were able to adapt with just minor modifications and some improvement. We were able to adapt the test to the new virus. So this virus this test, as I said, is an ELISA plate. So it can be run manually, or it runs on an automated platform like the Bio-Rad ones, of course, like the Evolis or the ATP, but it runs also on open platform. Any ELISA platform can run this test. So the assay has been launched end of April. it got the CE mark. It was registered on April 17th, and it's been submitted for EUA at the FDA. It's now under review by the FDA, and the registration is expected soon. So the use of this assay, as I said, is to detect the virus, the antibody against the virus, to detect the infection, and thus it is a very good, it's an aid to the diagnosis of the patients which present symptoms of infections, and that's a very good complement to the molecular methods, because as I said, you may have false negative away from the infection due to the fact that the viral load is decreasing. So in conjunction with the symptoms and molecular method, this test will help to diagnose the infection. And also, since it detects the immune response, it measures the immune response of an individual that was infected by the virus. It's a good way to determine the immune status of the person who are susceptible to have been infected. So this can be run at the individual level or population level to get an idea of the immune status of the whole population. So in terms of performances, we evaluate the sensitivity and the specificity of the assay. So as you can see, the sensitivity is quite high. We reached 97.5%. So meaning that we are detecting a very large number of the positive. The way we did it, we performed a longitudinal study, meaning that we have been following patients who were persons who were infected, and we have been following them over some time to detect their seroconversion. And this sensitivity, this very good sensitivity, demonstrates that we were able to track the seroconversion of all these patients over time. We also evaluated the specificity. The specificity is really key for this kind of ELISA assay because you don't want to have false positive, meaning that you want to be sure that when you get a positive, this is someone who has really been infected. And as you can see, the specificity is really very high with this assay. We reached 99.6% almost. And this has been done with a total of close to 700 specimens that were collected prior to the outbreak, meaning people who were not infected, and none of these samples turned out positive. So we have a very good specificity. And another key parameter for this kind of assay is the cross-reactivity, particularly with virus which are leading to a health condition close to the one led by COVID, like the flu, for instance. So we've been working with potentially interference specimen from different viruses like EBV, ZV, the flu, HCV, and none of them, no samples that were positive for this virus were detected by the antibody assay, meaning that there is no interferences with antibodies which are directed against those viruses. And another very key point was to check that we are not detecting other coronaviruses. So you know that coronaviruses are very common virus actually. So they can get you cold, for instance. The very common cold might be due to the coronaviruses. So it was important to avoid mixing those viruses with the COVID. So we've tested a positive specimen, meaning people who've been infected by this virus or co-infected by this virus and others, and who have developed immune response against them. And none of these samples turn out positive, meaning that there is no interferences with the antibodies that are raised against those viruses. So altogether, this data shows that we have a pretty sensitive and specific assay, very efficient to detect the antibodies against the COVID-19. Thank you very much. And I think now, Karine, I hand it to you for the Q&A sessions.
Great. Thank you. Thank you so much, Jean-Francois. Thank you, Monica. Thank you, Hestia, for presenting us with that information. I'm going to quickly reintroduce our panel and open it up for questions. We've had many, many questions coming in. It's been fantastic to see. So Monica Herrera, R&D Program Lead for Prenatal Testing and Infectious Disease at Bio-Rad. Hestia Mellert, Senior Director of Development at Biodesics. Jean-Francois Muscadet, Associate Vice President of R&D at Bio-Rad. Diana Marr, Applications Development at Bio-Rad. Jerry Bunya-Ratana-Kornkit, Principal Scientist at Bio-Rad. And Stephen Blakely, Global Director of Marketing for Gene Expression and Cloud at Bio-Rad. Okay, so our first question is for Hestia. Push the question here. Hestia, we're all thinking about heading back to work. Does Biodesics offer testing for pharma companies to test their returning employees? How about clinical trial patients?
Thank you, Carolyn. Yeah, this is a great question. Upon initial launch of the test, we had focused first in the hospital systems, but we are starting to engage and look at how we can support the community more generally and specific businesses of essential employees and employees that are trying to return back to the workplace. In addition, in terms of clinical trials, absolutely, we do have an active group that works with and supports pharma companies, and there have been multiple that have approached us with interest in using specifically the ddPCR approach for this testing. Some of the benefits of being able to do it by ddPCR, obviously, is the sensitivity of the assay as well as absolute quantification. And that's something that pharma companies seem to be really interested in and thinking that that will be useful for them.
Thank you. Our next question is for Jerry. How did you develop the controls that are used in Bio-Rad's test kit? What matrix did you use and what genes are in the controls? Jerry, can you pop off a mute and maybe tackle that question for our audience?
The first technical issue of the call. So thank you for telling me I'm on mute. So to answer that question, when we first developed this product, basically in February and launched the control in March, our product requirements are basically, we try to cover as many of the EUA assays that are out there. And so our strategy was to come up with in vitro transcripts, RNA transcripts for the virus that covered about 75% of the entire genome. And so as mentioned in the talk, not only does it cover the N gene, we also cover the E, the S, and the ORF1ab, which includes the RDRP gene. And we targeted 200,000 copies per ml. Every single one of these genes were value assigned by digital PCR. And we also included not in the standard as well as in the negative control, genomic DNA for the sample processing control. The matrix, as mentioned, is very similar to UTM, but includes additional stabilizers. And so far, we have tested this standard as well as the negative control across multiple EUA PCR assays, as well as the digital PCR assays that are out there.
Thank you. Fantastic. Next question is for Monica. Do you see the same issues with false negatives in your tests that are being reported on the news?
Thank you, Carolyn. That's a great question. And although no test can claim completely no false positives or false negatives, definitely if we go back to the limit of detection data that I showed, our test can detect very few viral copies per reaction. And I think that this is a key attribute for a highly sensitive test that would avoid high rates of false negative results. Now, something to keep in mind is that at this point, we don't have large data sets generated from patient specimens, but we are actively working internally at Bio-Rad and with our collaborators to test more and more patient specimens so that we can have specific claims on sensitivity and specificity.
Great, thank you. Hestia, the next question is for you. Actually, there are a couple of questions around the same topic, so I'm going to push one of them to the audience here and then mention a couple of the others that go along with it. So how many samples per day can your lab run? What's the turnaround time? And then there were some questions from the audience about what instrument configuration you have to achieve this rate of sample processing.
Thanks, Carolyn. Yeah, great question. Turnaround time is extremely important when you're, doing testing in this scenario. So I did describe earlier in that pie chart what the turnaround time looks like at this point. And that includes 70% of specimens getting out between 24 and 48 hours. A few specimens actually can get out the same day. We're trying to, compress that as much as we possibly can using the workflow. The nice thing about having, the setup in the lab, we have many readers, we have really trained staff. We can stagger the shifts, and that allows us to work 7 days a week at upwards of 1,000 specimens per day, so 7,000 specimens per week at this point. Currently, we are doing manual extraction. So something that we are definitely already scoping is implementing an extraction method that would be automated, and that could potentially compress that time and allow us to support higher throughput in the laboratory going forward. So that would be something that we would need to obviously validate and then implement in the lab in the future.
Great, thank you. Our next question is for Jean-Francois. There we go. Jean-Francois, given the problem with false positive results from some serology tests on the market, how does your test address this? And a few other questions have been coming in around the same kind of topic around specificity. If you could talk about specificity of your test as well.
Yeah, absolutely. So it's true that some very early tests showed defect in the specificity, meaning that they have a lot of false positive. So that's why we really took care of specificity with our assay. And as I said, the format of the assay, the sandwich format, is really the best one to minimize specificity. And as I showed on my last slide, we were able to reach a very, very high number of specificity. We are close to 100%, meaning that over more than 700 samples, I think that only one false positive showed up, and we are investigating why, but that means that we have close to no problem with the specificity with the assay.
Okay, great. And our next question is for Stephen. Here we go. Steve, will Bio-Rad develop an RT-qPCR test for COVID-19? And what things might I want to consider when deciding between qPCR and digital PCR?
Thanks, Carolyn. We are currently exploring the opportunity to develop an RT-qPCR assay from Bio-Rad. We have seen our platforms, our real-time PCR technologies adopted by many laboratories around the world for COVID-19 testing. So Bio-Rad is looking at a complementary solution for RT-qPCR with the digital PCR assay that has now been released. As far as choosing between RT-qPCR or digital PCR, there are different factors which can come into play, and one of them is a throughput. So with the real-time PCR assay being 1.5 to 2 hours, it lends itself to a much higher throughput. So when you're looking at screening or detecting a large volume of tests, RT-qPCR can be a good solution for you. Again, we see these as complementary technologies, and we're seeing laboratories around the world adopting the paradigm of real-time qPCR assays and digital PCR. When you're looking at low viral load or residual disease, digital PCR is certainly the good solution, as we've discussed here today, with the greater sensitivity.
Great, thanks, Steve. Next question is for Diana. The question, there you go, from our audience is, Why did we go with the Bio-Rad, go with the CDC sequences instead of the WHO or another design?
Hi, Carolyn. Thank you for that question. Yeah, so when we were trying to move really quickly to put an assay out on the market that we had good confidence in, we knew immediately that we wanted to use an assay that was already being used, a sequence that was already being used by other tests. So after reviewing all of the sequences, we chose the CDC assay because it had the shorter amplicon lengths, and which we know from ddPCR that can actually give you a lot of benefits, one of which is to be more sensitive, especially if you have a sample that is, been treated roughly, or if the RNAs has some breakage. So we went with that assay, and we've been quite pleased with its performance.
Very good. Thank you. Next question for Jean-Francois. Can you run the antibody test on existing ELISA systems, or does it need to be a Bio-Rad system?
No, it can be run on any open platform for ELISA assay. So of course, we prefer Bio-Rad's platform, and they are running the assay on our platform provide more safety and traceability because we have dedicated software, but it can be run on any open platform that will run plates.
Fantastic. Diana, another question from you, from our audience. Once the emergency is over, post-emergency, what kind of studies can the digital PCR platform enable?
Yeah, that's a great question. So that's something that, of course, we're already thinking about. One of the benefits that the digital PCR platform has right away is that it's quantitative by nature, and that there's no requirement for us to develop standard curve materials in order to get that quantitative answer from all of the assays that are put on the platform. We have gene expression assays, we have copy number assays, but one of the things that we see across the board with the cell and gene therapy market today is that viral titer is a very important benefit that you get from ddPCR. Is a very clean viral titer, and lab-to-lab day-to-day reproducibility will be critical for collaborating on a vaccine and vaccine development across the world. So we see our platform having a huge impact in speed and reliability of that kind of development post-emergency testing.
Great. I think we're all looking forward to the emergency being over. Thanks, Diana. The next question is actually for both Monica and Hestia. So maybe Monica could answer first and then Hestia. What kind of extraction kits are approved to run or you're using with your test? And then a few other questions sprinkled in throughout the panel here about, have you tried extraction-free or crude lysate or direct samples? So maybe each of you, Monica and Hestia, could talk a little bit about what extraction kits you're using in your configuration and what other elements of maybe extraction-free you've tried or have collaborators trying.
Yes, absolutely, Carolyn. So we have tried to give the broadest options for the labs when they run our test, and we have validated 2 extraction methods. One is a paramagnetic bead-based using Thermo Fisher's MagMax viral nucleic isolation kit. And the second one is a spin column or vacuum-based method using the QIAGEN viral mini extraction kit. We are certainly looking into extraction-less or extraction-free methods, and that is part of our phase 2. We have now submitted our EUA documentation, and we're trying to add on these additional supplements and improvements to the test. Hestia?
Thanks, Monica. I think the strategy at Biodesics is well aligned and similar to what you're doing. Obviously, there is always concerns about whether or not we're going to continue to be able to source the same extraction kit longer term. So we've started out using Zymo Research's kit, but also have experience, as Monica said, with the Qiagen and Thermo kits as well. And again, as we also not only are looking to stabilize inventory in this kind of uncertain time, but also trying to think about what kits make the most sense if we are to move to automation. So some kits are more amenable to being automated than others. And so we're really focused on understanding that. The magnetic bead kits, for example, like the Thermo one Monica mentioned, would be a really nice transition into an extraction method that was automated, for an example.
Okay, the next question, I'm going to push just one of the audience members' question, but there were a lot of questions coming in around the sample type, specifically the nasal swabs, and then what Bio-Rad or Biodesics has maybe looked at in terms of saliva as a sample type. So maybe, again, each of you want to take that as a question, and then we'll have another question here in a second for Jean-Francois about different blood collection as well.
Sure, maybe I can start that one off. So we initially did our validation with the NP swabs, as I had mentioned earlier. But yes, there has been a lot of excitement, both in the field as well as at Biodesics, regarding saliva. Obviously, it opens up a lot of opportunities and a much perhaps easier way of actually collecting a patient's specimen. I think that there has been some mixed data, and FDA seems to be really keeping a close eye on this and understanding what data people have to support the validation of their saliva tests. Of course, Rutgers recently was one of the first to actually show data that was up to the standard that would be required and has gotten the EUA for that test. So yeah, we're definitely pursuing it at Biodesics, interested to see and keep track of where the field is going, and hope to have a saliva test as well as an option in the future.
And to add to what Hestia just said, it's similar for our intents at Bio-Rad. We definitely see that saliva is an ideal sample type in terms of ease of collection. And as Hestia mentioned, there have been some conflicting reports as to what amount of virus can be detected from saliva compared to nasopharyngeal swabs or other respiratory sample types. So our intent also is to start collecting the saliva samples and do paired testing with nasopharyngeal swabs and saliva samples to see what the performance is for saliva as a sample type, so that we can submit to FDA. And we're also looking forward to generating the data with the performance that FDA expects.
Great. Next question, Jean-Francois, is for you. There are several questions from the audience around why your approach uses total antibodies and not an IgM or IgG. Does it give the same information? Can you talk a little bit about the decision for that?
Okay, so the decision for total antibodies is really linked to the sensitivity. It's clear that by adding both IgG and IgM, we are really able to increase the sensitivity of the assay. Reported in the literature for other assays, so this seems to be a general observation for this kind of assay. So since we wanted really to be complementary to molecular method and to be able to detect infection as early as we can, we chose to use the total antibody. That's the first reason. Other reason is that at this time now, there is no very strong knowledge about the kinetics of the immune response. There are a few studies out there, but so far we don't know exactly when the IgM and IgG are rising up and how long they would stay, how robust is their immune response, IgM, IgG. You know that usually you have a very early response with the IgM, and then IgG are following, and they are telltale of a robust immune response over time. So this is what is expected, but we don't have this knowledge at this time. So betting on IgM or IgG now that we are still at the heart of the pandemic was kind of a guess. So we decided to go for the total antibody to make sure that we have the best sensitivity and we will get as many positive as we can. So that's why we chose total antibody. So it's true that maybe later on, when we will try to get some general idea about, for instance, the immune status of the population, IgG might be a good test, but so far we don't have enough knowledge about that. So if I had to give you a guess, I'd say that probably having a total antibody first and then an IgG assay might be the good assays on the long term. But as for now, the total antibody is probably the best kind of assay that we can have.
Okay, the next question from our audience is around environmental and soil samples. So let me push that here. So a question from the audience: we're intending to use this for environmental monitoring, therefore surface swabs. Since we're not intending for clinical, can we use standard one-step reagents such as the TACPath or Luna mixes? And another question that came in related to this was, have we done any work detecting in the soil? So I'm not sure if, Monica, if you want to take that one?
So we have not tested anything outside human specimens. Of course, used as a research use only tool, our assay can be used for testing environmental samples. The one-step ddPCR reagent and in the workflow that we described would work. Diana, is there anything else you would want to add to that?
Yeah, Monica. So the only thing that I would add to that is this question about using other super mixes inside our droplet technology. Unfortunately, if that's this time, you cannot do that. You must use our reagents because they can both produce uniform droplets as well as the reaction inside of each of the droplets. So they've been optimized. And so you will have to use our Bio-Rad Droplet Digital PCR one-step reagents.
Great, thank you. Question, Hestia, for you. Can you please clarify the samples you're testing are from patients exhibiting clinical symptoms or non-clinical patients? I think maybe they mean non-symptomatic patients.
Yeah, right. Good question. So currently, the specimens that are coming in are from hospital systems. So there are patients that have most generally have symptoms or are suspected of having COVID-19. So currently that is who we are testing, although we do are scoping and looking at how we can support the community more generally, even in populations where patients or people may not be symptomatic.
Question for Monica. How do you report results, in copies or just as positive and negative? There were actually several questions from our audience members about quantitative, qualitative, how we're reporting these results.
That is a fantastic question. And I may not be able to give a definitive answer since we are still in ongoing discussions with FDA, and FDA is reviewing our submission. However, currently, as we have described our intended use, this is a qualitative test where the test result is determined as detected or not detected for the SARS-CoV-2. However, similar to RT-qPCR, we do have thresholds and cutoffs that are based on concentration, so copies per microliter, and number of positive droplets. Right now, the cutoff that we are recommending is greater than or equal to 2 positive droplets and greater than 0.1 copies per microliter. But again, as I mentioned, this is still preliminary and under review from FDA.
Great. Next question, Jerry, is for you. The question is, if our exact diagnostic standards contain guanidine thiocyanate or other elements that could inhibit the enzyme's activity?
No, it does not contain any guanidine thiocyanate. The majority of, as I mentioned, it's very similar to universal transport media. There's some carrier RNA in there to stabilize the in vitro transcripts, but it should not have anything that inhibits enzyme activity.
Next question, Jean-Francois, similar to the question that Monica just answered a moment ago, how are you reporting your serology results? Positives or negatives, or some more quantitative result?
No, positive and negative, definitely. That's the way the result is expressed is a ratio of the signal over a threshold, which is called the cutoff, which really gives you whether this is positive or negative. So you get this result and you get an idea of whether this is a strong signal or a weak signal, but this is not really quantitative. This is called semi-quantitative, because you can get an idea from these results of whether you have a very strong immune response or a weaker one, but this is not really quantitative.
Great. And then our final question, since we're almost to the top of the hour here, is for Monica. And the question from our audience is, what is your minimal viral copy detection rate? So what is our limit of detection in the digital PCR test?
Yes. So as I mentioned, our preliminary data, and again, with the big asterisk that this is still under FDA review, our results show that we can detect down to 5 copies per ddPCR reaction with a detection rate of greater than or equal to 95%, which makes this our limit of detection.
Great. And I think we have time for just one last question for Jean-Francois. You presented the antibody test. Do we have a test for the antigen in development or ready to release, and is it scalable? So anything on the antigen?
We are thinking about it. We don't have one right now, and we are not sure at this time that we are going to develop. We are just this is in our mind, and we haven't decided yet.
Okay. Great, thank you. Well, that's all the questions we have time for today. There are still, I see 109 unanswered questions in our queue here. Thank you to our panel and our audience for your participation. We didn't get to everybody's questions, but we will make sure that people from Bio-Rad follow up with you directly in the next couple of weeks to answer all those questions. As a reminder, today's webinar will be available on demand within 24 hours. Be sure to check out the items in the resource list on your screen or visit us online at biorad.com for more information. Thank you, everyone. Have a great rest of your day and stay safe.